everyone, today I'll be talking about metabolic versatility of E. coli. In order to produce energy to live, organisms must produce ATP. Prokaryotes like E. coli produce the most ATP via aerobic respiration using oxygen as a final electron acceptor. As a brief overview of aerobic respiration, one molecule of glucose is broken down via glycolysis and converted into pyruvic acid. This process produces a net 2 ATP and 2 NADH, which is a reduced electron carrier. The pyruvic acid goes through the Krebs cycle, where 2 more ATP is produced, 2 FADH2, which is another electron carrier, 8 NADH, and 6 molecules of carbon dioxide. During the next phase of cellular respiration, a series of reductions indirectly transfers energy from NADH and FADH2 to ATP during the electron transfer chain. The ETC consists of a series of membrane-bound carrier molecules that pass electrons from one to another, which are capable of oxidation and reduction. As electrons are passed through the chain, there occurs a stepwise release of energy. In prokaryotes, it is found in the plasma membrane. The ETC starts with NADH oxidation and regenerates NAD+. The electrons are then transferred through a series of membrane-bound carrier molecules and ultimately to the terminal electron acceptor, in this case oxygen. Energy from electrons is used to actively pump protons across the membrane, thereby establishing a proton gradient that generates ATP by going through ATP synthase. In the end, 38 ATP are generated. If oxygen isn't around, E. coli can also use alternative electron acceptors and undergo anaerobic respiration. Nitrate, or NO3, is one of the most common electron acceptors and can be reduced to nitrate, or NO2. Some organisms can also use sulfate as a final electron acceptor to form hydrogen sulfide. Bacteria using nitrate and sulfate as final electron acceptors is essential for the nitrogen and sulfur cycles that occur in nature. Although E. coli does not do this, other bacteria are capable of further reducing nitrate into nitrogen gas through a process called denitrification. Nevertheless, the process is very similar to aerobic respiration, except that nitrate is the final electron acceptor, and this process produces less than 38 ATP. Finally, E. coli can also undergo fermentation. Fermentation can occur in the presence of oxygen, but is not required. Fermentation also does not require the Krebs cycle or ETC. An organic metabolite like pyruvate is used as a final electron acceptor. It's also used to regenerate NAD+ from NADH and generates 2 ATP. E. coli specifically performs ethanol fermentation, and other organisms like yeast also do this and is used in alcoholic beverages. The metabolic versatility of E. coli is amazing, and thank you for listening.